Hello students, welcome to this new session on DNA microarrays. So DNA microarray is a very powerful tool for the analysis of whole transcriptome in transcriptomics and expression profiling. So array means orderly arrangement of objects or the things. So in case of DNA microarray, we arrange single-stranded oligonucleotides on a very small surface area of the glass slide, and that is why it is called as DNA microarrays. So the contents of this lecture are introduction to DNA microarray, its brief history, then the principle of DNA microarray technology, that how it works, how it functions, then two types of DNA microarrays we are going to discuss in detail, that is the glass cDNA microarrays and in situ oligonucleotide array formats. And finally, we will discuss applications of microarray technology. Introduction to DNA microarray. So with the advancement of DNA sequencing techniques, we come to know about the whole genome sequences of many prokaryote as well as eukaryotic organisms. Along with that, now we are able to immobilize thousands of single-stranded oligonucleotide DNA sequences on a very small surface of a glass slide or a membrane. So this technique has led to the development of microarray technology. So in DNA microarray, single-stranded DNA fragments, they are coated, they are immobilized on a very small surface of a glass slide, which is called as a chip or a slide. Now the single-stranded DNA molecules that are coated on the glass slide, they are called as probes. Then this microarray technology, it make use of a hybridization to detect the specific DNA or RNA present in the sample. So you isolate your DNA or RNA from the sample, then you label them with some kind of reporter dye, and then you allow these single-stranded DNA or RNA molecule to hybridize with the probe which are coated on the glass slide. So this DNA microarray uses millions of different probes which are fixed on the solid surface of the glass. What is an array? Now the meaning of array is orderly arrangement of objects or the things. In case of DNA microarray, it is an orderly arrangement of samples where matching of known and unknown DNA sample is done based on the base pairing rule. In case of DNA microarray, the known DNA sample is probe, which is coated on the glass slide, and the unknown DNA sample is the sample which is isolated from the cell and it is labeled with some kind of fluorescent or a reporter dye. Then you hybridize this known and unknown DNA sample on the basis of base pairing rule, and then you observe the results. In an array experiment, it make use of a common assay system, such as a microplate or a standard blotting membrane. So a DNA microarray can also be set up on nitrocellulose membrane or a nylon membrane, or it can also be set up on the glass slide. So in this picture, you can see a robotic arm with the spotting slide. So this robotic arm simply work as an inkjet printer, where the oligonucleotide single-stranded DNA molecules are coated on the glass slide in the predefined manner. History of DNA microarray technology. So microarray technology is evolved from Southern blotting. In Southern blotting, we detect the presence of specific DNA sequence present in the sample by using single-stranded chemically synthesized probe molecules which are attached with radioactive material or a fluorescent dye. Now in case of Southern blotting, the probe molecules are free and the DNA molecule from the sample is immobilized on the nitrocellulose membrane. But in case of microarray technology, the probe molecules are immobilized on the glass slides and the sample DNA, which is isolated from the cells, it is free and it is labeled with some kind of radioactive material or fluorescent dye. Now the concept of microarray was first proposed in late 1980s by Ogenlich and his colleagues. They spotted 4,000 cDNA sequences on nitrocellulose membrane and used radioactive labeling to analyze differences in gene expression patterns among different types of colon tumors in various stages of malignancy. So they isolate mRNAs from the cell, they convert them into cDNA. Those cDNA molecules were spotted on the nitrocellulose membrane, and later on the mRNA molecules isolated from the different stages of the malignancy of the colon tumor, they were labeled with radioactive material, and they were hybridized with this 4,000 different cDNA molecules, and thereby they studied the gene expression pattern among these different types of colon tumors in various stages of malignancy. Principle of DNA microarray technology. The core principle of DNA microarray technology is hybridization between the two DNA strands. As you can see in this picture, single-stranded probe molecules are fixed on this glass slide, and then they are mixed with labeled targets, that is, the RNA or DNA molecules isolated from the sample and labeled with reporter dye. Now the DNA strands from the sample which have full complementary with the probe, they will bind strongly, and the labeled target from the sample, those which have partial complementary to the probe, they will bind weakly. Now the fluorescent labeled target sequence that bind to the probe sequence generate a signal that depends on the strength of hybridization determined by number of paired bases. So if the DNA from the sample, it forms full complementary base pairing with the probe sequence, then the intensity of the fluorescence will be high. And if only partial complementarity is present in between the sample and the probe, then the intensity of the fluorescence will be less. The DNA microarray technology is a high throughput and versatile method which is used for parallel gene expression analysis for thousands of genes of known and unknown function. So by this method, we can analyze the whole transcriptome of the cell, that which genes have been upregulated or which genes have been downregulated. And this method is used for the detection of polymorphism and mutations in the genomic DNA. So nowadays this DNA microarray technology can be used for the detection of single nucleotide polymorphisms present in the genomic DNA among the population. A DNA microarray is a collection of microscopic DNA spots on solid surface. Each spot contains picomoles of specific DNA sequence known as probe or a reporter. So in this DNA microarray technology, on the glass slide, the area is around 1.5 centimeters square, and in that area, the single-stranded DNA molecules are coated or immobilized, and those single-stranded DNA molecules coated or immobilized are known as probes or the reporters. So each single-stranded probe DNA molecule that is coated on the glass or a silicon chip or a nylon membrane, it corresponds to the fragment of genomic DNA, cDNA, PCR products, or chemically synthesized oligonucleotide, and it represents a single gene. So from the genomic DNA, the known gene sequences are used for the synthesis of cDNA, or they may be amplified with the help of PCR, or they are chemically synthesized. So these single-stranded probe DNA molecules representing all the genes present in the genome, they are coated on the glass slide or a silicon chip. Then the probe target hybridization is detected. Now in this case, the target DNA, which is isolated from the sample, it is actually labeled with some kind of fluorescent dye or other reporter molecule. So this probe target hybridization is usually detected and quantified by detection of fluorophore, silver, or chemiluminescence labeled targets to determine relative abundance of nucleic acid sequences present in the target. Then the principle of DNA microarray technology. Now this principle is dependent on the hybridization of two complementary DNA strands. One DNA strand that is immobilized on the glass slide, which is called as probe DNA molecule, and another single-stranded DNA molecule which is isolated from the sample, which is called as a target. Now these two DNA molecules, if are complementary to each other, they will form base pairing, and that is the principle of DNA microarray technology. So there are four major steps in performing typical microarray experiments. The first step is sample preparation and labeling. Now in this step, we isolate DNA or RNA from the cell sample. Then we convert them into single-stranded form and we label them with some kind of reporter molecule. Then these target DNA molecules along with the label, they are mixed with the probe molecules which are coated on the glass slide, and thereby after forming the complementary base pairing, they will hybridize with each other. The next step is washing. Now in the washing step, the target DNA molecules which do not attach or which do not form complementary base pairing with the probe, they will wash away. And finally, image acquisition and data analysis. Later on, the glass slide is exposed to the laser light, which will excite the fluorescent dye, and it will emit the higher wavelength light, which will be detected by the detector, and the image will be formed. So that image is acquired with the help of confocal microscopy, and later on that image is analyzed. That is called as data analysis. The first step in DNA microarray technology is sample preparation and its labeling. First of all, from the control as well as from the treated cell sample, total RNA is extracted. This total RNA also contains mRNA molecules, which represent the quantitative copy of genes expressed at the time of sample collection. Then this collected RNA molecules are converted into the cDNA molecule by the process of reverse transcription. Some short primers are required for this process. Now each cDNA molecule from both the samples, that is from control as well as non-control, that is from maybe treated and untreated, it is labeled with the fluorescent dyes. The dyes that are used in this technique are sign-in dyes, that is Psi3 and Psi5. Now Psi3 has an excitation wavelength of around 550 nanometer and emission wavelength of 570 nanometer, which is green in color. And Psi5 has excitation wavelength of 650 nanometer and emission wavelength of 670 nanometer, which is red in color. So in this diagram, you can see that RNA is extracted from two samples, that is control as well as non-control. You can call non-control as a treated or a treatment. Now this RNA extracted from both the samples converted into cDNA by the process of reverse transcription, and the cDNA molecules collected from control cells, they are labeled with Psi3 dye, and the cDNA collected from non-control cells, they are labeled with Psi5 dyes. Later on, both of these cDNA molecules are mixed together and they are applied on this glass slide for hybridization with the probe molecules. Then array hybridization. Now here the labeled DNA, that is cDNA, that is from sample as well as control, they are mixed together. After mixing, they are purified, and after purification, the mix labeled cDNA is competitively hybridized against denatured PCR product or cDNA molecule spotted on the glass slide. So already the PCR products or cDNA molecule which are representing the gene sequences, they are immobilized on the glass surface. They are called as probes, and to these probes, this purified mix label cDNA is applied for the hybridization. Now wherever there is a complementary base pairing, this cDNA will hybridize or form complementary base pairing with the probe molecule. Then image acquisition and data analysis. Now the slide where hybridization was carried out is dried and it is scanned to determine how much labeled cDNA is bound to each target spot. So generally this scanning is done with the help of confocal microscopy having fluorescence attachment, and the image is acquired for further analysis. Then hybridized target produces emission. Remember, only where the target DNA molecule hybridizes with the probe, there only you will get the fluorescence emission. Wherever the single-stranded probe is unoccupied or it is not forming complementary base pairing with any of the target, there no emission will occur. That spot will remain black in color. Then later on, microarray software is used for the analysis of this image. Now in this picture you can see gene chip showing different types of color spots. Now here you can see three colors, that is yellow, red and green. Remember, The green color dye we use in the control sample, in the control, and red color dye we use in the non-control or the treated sample. Let's suppose that in our example, the control is the normal cell and the non-control is a cancerous cell. So you isolate mRNAs from control, that is from non-cancerous cells or normal cells, and you isolate mRNAs from the cancerous cells. Then you label the cDNA molecules from the control with the green color dye, and you label the cDNA molecules from the cancerous cells with the red color dye. Now here you can see some spots are green in color. It indicates that this gene was only expressed in the normal cell. It means that this gene's expression is strongly suppressed in the cancerous cells or in the sample. Understand? Because these genes have only expressed in the control and they are not expressed in the non-control. It means that these genes are expressed in the normal cells and they are not expressed in the cancerous cells. So this means that this gene expression is suppressed in the cancerous cell. On the other hand, some spots are red. Now this red spot means this gene is only expressed in the cancerous cell and it does not express in the control cells or the normal cells, which indicate that the gene expression is induced in the cancerous cell, which is not there in the normal cells. And some spots are yellow. Now this represent that there is no effect on this gene expression in control or normal or cancerous cell line. The gene expression is equally abundant in both the samples, that is in the normal cell line as well as in the cancerous cell line. So in this way we can read the results on this image of DNA microarray with the help of these color spots. Then next we are going to discuss about the types of DNA microarray. Now there are two types of DNA microarray. One is glass cDNA microarray, which involve the microspotting of prefabricated cDNA fragments on the glass slide. And the second one is high density oligonucleotide microarray, often referred to as CHIP, which involve in situ oligonucleotide synthesis. The first type is glass cDNA microarray. This glass cDNA microarray was the first type of DNA microarray technology which was developed, and it was pioneered by Patrick Brown and his colleague at Stanford University. It is produced by using robotic device which deposits a nanoliter of DNA onto the coated microscopic glass slide around 50 to 150 micrometer in the diameter. As you can see in this picture, it is a contact printer with robotic pins. So this is actually resembles the head of inkjet printer, and through these needles or through these pins, prefabricated cDNA molecules or PCR products or chemically synthesized oligonucleotide sequences, they are coated on the surface of this glass slide. The glass slide is previously coated with the polylysine so that covalent bonding in between this DNA molecules and that polylysine is possible. So in this way, glass cDNA microarray is produced. Then the manufacturing of glass cDNA microarray. So this is the detailed protocol of manufacturing. First of all, you have to select the material for spotting onto microscope glass surface. Selection of material means whether you want to use cDNA molecules or PCR product or chemically synthesized oligonucleotide sequences. So after selection of the material, you have to prepare it. So maybe you have to go for synthesis of cDNA molecules or amplification of DNA with the help of PCR or chemical synthesis of DNA. After preparation, purify that material which is representing the gene of interest, and finally that DNA molecule is spotted onto chemically modified glass slides via contact printing or inkjet printer. So this is the spotting of the slide you can see in the picture here. This is the printing head and this is the master plate. So through this master plate, the prepared sample, that is the DNA molecule, is used. This is a wash solution for the washing purpose, and these are the slides where this printer will coat the probe DNA on the surface of the glass slide. So this is how the manufacturing of glass cDNA microarray is carried out. Next is advantages of glass cDNA microarrays. Now the first very important advantage of this method is it is very cheap. It is lower in the cost. So many of the laboratories are able to afford this glass cDNA microarray because of its lower cost. Next is its accessibility. Require no specific equipment for use, such as hybridization does not need any kind of specialized equipment. You can simply add the target DNA solution on the glass slide and it will be hybridized. You don't need any special equipment for the hybridization purpose. And finally, data capture. Now, the data capture can be carried out by using equipment that is very often already available in the laboratory. So here you can use the fluorescence microscope also for the capturing of the data, which is usually available in many of the laboratories working in the field of molecular biology. Then disadvantages of glass cDNA microarray. It has few disadvantages such as intensive labor requirement for synthesizing, purifying and storing DNA solution before microarray fabrication. So for this glass cDNA microarray, you have to prepare the cDNA molecules, or you have to amplify the DNA with the help of PCR, or you have to chemically synthesize the DNA molecule. And that is what a very labor intensive job, that synthesizing, purifying and storing the DNA solution. So that is one of the disadvantage of glass cDNA microarray. Then they may hybridize to spots designed to detect transcript from the different gene. So that may happen that the molecule that you have prepared for the glass cDNA microarray, it may hybridize to another spot that is actually used to detect the transcript from the different gene. Then second type of microarray is in situ oligonucleotide array format. It is called as in situ because the single-stranded DNA probes are actually synthesized on the glass chip itself. So oligonucleotides are synthesized on the chip. Presently, a commercial version of it is available that is called as Affymetrix gene chip. Now these chips can hold around 5 lakh probes per site in the area of 1.28 centimeters square. And due to this very high information content, they are finding widespread use in hybridization based detection and analysis of mutation and polymorphism, such as single nucleotide polymorphism. Now, these ready-made Affymetrix gene chips are available for many of the species. On the single Affymetrix gene chip, you may find genes of whole organism, or the genes representing the whole genome of an organism. So the Affymetrix gene chip, it has very high information content on it. The next is manufacturing or synthesis of in situ oligonucleotide array. Now this process is light directed, and that's why called as photolithography process. Now here you can see in this diagram, the glass slide is coated with some kind of organic material like polylysine, where nucleotides can be covalently attached. But the functional groups are coated with photolabile protecting group X, called as MENPOC. It's a organic chemical which is photolabile. So whenever the light hits this photolabile protecting group, it will be removed and functional group will be exposed. Now here you can see in step A, the organic molecule functional groups are not exposed, they are attached with photolabile protecting group, and then we apply the mask. Now mask is applied at the places where we do not want the photolabile protecting group to be exposed to the light. So here in this example you can see here this is the black color mask and this is the black color mask. So these protecting groups will not be removed even after exposure of the light because they are masked. And this second and third group will be exposed to the light because mask is absent there. So once the light hit these two protecting group, they will be removed and functional group will be exposed. That is OH group will be exposed there. Later on, you add the nucleotide which you want to add at that particular site. So in this case, it is adenine, that is A. Now, these nucleotides are also attached with photolabile protecting group X. Okay. So as you can see here, the A is attached at the desired site. Then in the next step, again the mask is changed and mask is applied at the position where you don't want to remove the photolabile protecting group, and positions are open somewhere where you want to remove the photolabile protecting group. Then you add next nucleotide. In this case, it is T. So T is added here. Again, T is attached with the photolabile protecting group. So after light is hitting these two positions, the photolabile protecting group will be removed and T will be attached at those two positions. Okay. Then you repeat this procedure, and by repeatedly masking and exposure to the light, you can synthesize the desired oligonucleotide sequence. So activation of areas for chemical coupling, attachment of nucleotide containing photolabile protecting group is carried out. Next, light is directed to the different region of the array surface through a new mask. Then addition of the second block of T containing a photolabile protecting group X, and this process is repeated until the desired product is obtained. So in this way, in situ oligonucleotide arrays are synthesized, or Affymetrix gene chips are produced. Then the advantages of this in situ oligonucleotide array format. The very important advantage is that it is very fast, that is its speed, its specificity and reproducibility. So these are the three advantages. As compared to glass light, these are very fast, they are specific, and the results are reproducible. Then the disadvantages. The major disadvantage of this method is expensive specialized equipment. For example, to carry out hybridization, staining of label, washing, and quantitation process. Then short sequences used on the array have decreased sensitivity binding compared with the glass cDNA microarray. So these are two disadvantages of this method. Then applications of microarray technology. Now, there are several applications of DNA microarray technology. Few of them are like it can be used as gene expression profiling tool, or as a comparative genomics tool, or disease diagnosis, for drug discovery, for toxicological research, etc. So first application is microarray as gene expression profiling tool. Now, the principal aim of using microarray technology as a gene expression profiling tool is to answer some fundamental questions in biology, such as when, where, and to what magnitude genes of interest are expressed. So, as I have already told you that DNA microarray technology can be used for the study of transcriptome. So, suppose you have two different set of samples and you want to see that how the gene expression pattern is changing when the particular gene is expressed, in which conditions it is expressed, and up to what extent or up to what magnitude it is expressing. So in order to analyze all these things, we can use DNA microarray technology. Then microarray analysis measure changes in multi-gene pattern of expression to better understand about regulatory mechanism and broader bioactivity function of the gene. Then microarray as comparative genomics tool. So, you know that in the population, there are single nucleotide polymorphism or polymorphic DNA is present. And in order to analyze or to compare this polymorphic DNA, we can use DNA microarray technology. So it has widespread use in comparative gene mutation analysis to analyze genomic alterations such as sequence and single nucleotide polymorphism. And in microbiology microarray, gene mutation analysis is directed to characterization of genetic differences among microbial isolates, particularly closely related species. So many times the morphological characteristics of two different species may be similar, and that is why they are not recognized as two different species. So we can make use of this DNA microarray technology for characterization of genetic differences among those isolates, and we can prove them as two different species. Then for disease diagnosis. Now, different types of cancers have been classified on the basis of the organs in which the tumors are developed. Now, with the evolution of microarray technology, it will be possible for researcher to further classify the types of cancer on the basis of pattern of gene activity in the tumor cell. So we know that there are different types of cancers on the basis of organs in which they are arising. Like there are carcinomas, there are sarcomas, leukemia is there. But if the cancer is present in the single organ, but in among different individual, the gene expression pattern is different. Like suppose there is a colon cancer in different individual, but the gene expression pattern is different in among those individuals. Now that kind of study can be done with the help of this microarray technology for the further classification of the types of cancer present in the single type of organ. Then drug discovery. So microarray technology has extensive application in pharmacogenomics. Like comparative analysis of genes from disease and a normal cell will help in the identification of biochemical constitution of the protein synthesized by the disease cell. As in the case of microarray example, we discussed that how we can identify that which genes were upregulated or downregulated in the normal cells and cancerous cells. So that can be done with the help of this. Then application in toxicological research. So whenever the cell is exposed to the particular type of toxin, the gene expression pattern changes. So in order to understand that how a cell respond to the particular toxin, we can use this DNA microarray technology. So toxicogenomics establishes correlation between the response to the toxicant and the changes in the genetic profile of the cell exposed to such toxicant. The microarray permits researcher to examine thousands of different genes in same experiment and thus to obtain a good understanding of relative level of expression between different genes in an organism. So again, like in case of cancer cells, you can take two different samples. That is, one is control and other is treated. In control, you do not expose the cells to any kind of toxicant, and in the sample or in the test, you expose them to the toxin molecule, and then you analyze that how the gene expression pattern is changing after exposure to the toxin, and thereby we can analyze thousands of genes at a time, and we can find out the candidate genes which are responsible to the uh, response against the toxin. So conclusion: Microarrays recently developed functional genomics technology that has powerful application in wide array of biological, medical sciences, agriculture, biotechnology, and environmental studies. Since many universities, research institutes, and industries have established microarray-based core facilities and services, microarray have become a readily accessible, widely used technology for investigating biological systems. So that's all about the DNA microarray technology. Thank you.